This is just going to be talking about some of the cell signaling methods that are happening in our bodies, and then just a little bit at the end about drug development and creating drugs for various diseases. There's different types of signaling that we can talk about. Juxtacrine is signaling from something just right to cells, so these little blue boxes are supposed to represent cells. And juxtacrine signaling would be just cell-cell interaction. So usually something that's attached to the cell or on the cell surface, and it's not releasing it or excreting it. It's just a cell touching a cell, so that would be called juxtacrine. Autocrine is when that cell signal is actually, it's being released from the cell, but then it's actually being received by that same cell. Sometimes that does happen. That's the autocrine type of signaling. Paracrine is when it actually goes out and reaches other cells from a short distance, maybe 15 or so cell diameters or 20 to 200 microns away from it. So that's paracrine. And endocrine is when we're talking about dumping that stuff into the blood or other systems so that it can transport further, the signal can transport. So these are all induced, these factors are all considered hormones. So when it's sending a signal away from itself or to another cell, unless it's autocrine obviously, then they're referred to as hormones. Now these hormones don't carry out chemical reactions. They're not part of that chemical reactions. They're simply messengers that tell another cell to do something and change its biochemistry in the cell. There are three major types or classes of hormones that we'll talk about in the endocrine system. First is amino acid derivatives. So you can see that melatonin is an amino acid derivative. And so this is a smaller compound. But if it's an amino acid derivative or an amino acid, another hormone, or it's actually neurotransmitter is glutamine or glutamate, those are other types of hormonal signaling molecules. So really, as we learn more about neural processing and hormonal cellular signaling, there's really not that big of a difference between how neurotransmitters work and how regular cell signaling works. So it's just in a different part of the body. But anything that's an amino acid or looks like an amino acid is the first type here. Amino acids or amino acid derivatives. Next type are polypeptides. So you'll have multiple amino acids bound to each other, but usually less than 20 amino acids. So here vasopressin is a polypeptide that controls urine volume. We're talking about 2, 4, 6, 8, 9, 9 different amino acids. We have the N-terminal and the C-terminal N that's been modified, right? So it's still got that amide functional group on here. So this is these are smaller peptides, and the last one are steroids. So these steroid compounds are basically, we're looking at the cholesterol. Remember, we talked about the synthesis of cholesterol. So anything that looks kind of like this. This is an estradiol. It's derived from cholesterol, and it's an estrogen type compound. So those are the three different types. So this one's easy to recognize, the polypeptides, and this one basically, if it doesn't look like cholesterol or a polypeptide, so if I'd ask you a question about that, it's an amino acid. It's derived from an amino acid. I'm not going to spend too much time on this slide, but just briefly to describe where these things are coming from. There are different sites or sources for hormonal signaling. So adrenal cortex, testes or ovaries, right? Different glands within your body will release these. Examples of steroid hormones are cortisol and estrogen, progesterone. So these things will regulate your stress levels, inflammation levels, glucose metabolism, sexual characteristics. For example, amino acid derivatives, a couple of them, epinephrine, norepinephrine, going to regulate glucose metabolism, blood pressure, heart rate. Thyroxine is a general controller from your thyroid glands, going to control general growth and development. And finally, some polypeptide hormones. So we have multiple your pituitary glands are releasing some of these growth hormones, follicle stimulating hormones, thyrotropin, for example. So all of these different regions, and they might have different targets, right? So even though they might be released from a single gland, they might have different peripheral targets. Just a little bit about where these hormones might be coming from. But I'm more interested in talking about the biochemistry and what is happening when these hormones get to the cell. So one of the things that I mentioned, or that you should recognize, is that steroid hormones are made from cholesterol. We know that cholesterol is fat soluble. It likes to get into the membranes of our cells, and it can also, therefore, because of that, get through the membranes. There's no transport that needs to happen. It's a passive activity. It can diffuse through the cell wall and get into internal cytoplasmic receptors. So steroidal hormones usually work through it just comes right through the membrane and receives its receptor in the cytosol of the target cell. Polypeptide derivatives or amino acid derivatives, they are going to be received by a membrane receptor, something outside the cell, and then they're going to send a signal into the cell in another way. We're going to talk about this signaling mechanism over here. We're not going to talk as much about the or the steroid hormones because these just kind of bind to the receptor and generally then change the gene activity directly. So once bound, they get into the nucleus, and then they activate transcription of certain sets of genes. But these pathways, we're going to focus more on the receptors and how they are activated. One of the common types of mechanisms involves a G protein complex or GTP complex, and which activates a second messenger, a secondary messenger. That means a messenger inside the cell through ATP that turns into cyclic AMP. So any hormone, either peptide derived or amino acid derived, that will bind to the receptor. Once bound, it actually recruits and it has a higher affinity for this GDP compound. So it becomes active, it binds, and then this GDP compound becomes active and it binds to ATP. So this is the active GTP enzyme. And then once this has become activated, then it actually will bind to the adenylate cyclase compound that will activate the formation of a ATP to cyclic AMP. So this is the response in the cell. So once the cyclic AMP is formed, that's the second messenger that goes on and activates more signaling cascades. So this is an example of what this is. The ATP, once the adenylate cyclase actually will kind of create this circular, so that's why it's the cyclase. It actually makes this ring of the phosphate group here, and this is the second messenger that will then go on to interact with other things, be bound by other proteins and enzymes that will activate signaling cascades. This can be hydrolyzed and broken. The ring can be broken, and then we can reform AMP. There are a lot of different types and classes of families based on those secondary messengers and the receptor for that signaling factor or that hormone. I'm going to go through a couple of these. I spend a lot more time in developmental biology looking at signaling transduction mechanisms. So if you're interested in that kind of thing, we talk about it a little more in that class that I'll be teaching next semester or next year. But for now, I'm going to just focus on a couple of these, and one of them I'm going to look at right now for you is the FGF signaling mechanism, and that's because we go through this tyrosine kinase pathway. Phosphorylation, here we can see that this actually has two different receptors. That ligand or the paracrine factor that is bound is received, and it actually brings together these two different cellular compounds. So you see these alpha helices within the cell membrane. That's what holds it in the membrane. But it's not until one of these binds that it actually brings these two things together. So once they're together, it's kind of cool because it activates, this acts as an enzyme that together, bound to itself, it autophosphorylates itself. So the phosphorylation happens internally, and that eventually leads to active transcription of a specific set of genes within the cell. But this binding brings these together, activates the phosphorylation, and then we actually hydrolyze. We use ATP. There's a transferase reaction. This kind of becomes an enzyme, right? A kinase that activates a protein. So we phosphorylate a protein, and it changes the shape of this protein, and this can go on and on and on and on. So here's another example of this. This is a receptor tyrosine kinase signaling transduction pathway. Similar idea. We have two components. And then the ligand brings these two together, and then we get a bunch of phosphorylation events. So these get really complicated. People have spent many years mapping out exactly which compounds get phosphorylated, where they happen, and then that phosphorylated enzyme or compound can then recruit the attachment of different things. So we can see here, this example, the ligand comes and binds to the receptor tyrosine kinase. That activates phosphorylation on that kinase. Then we can recruit the GTP exchange factor, GEF, that can then interact with RAS, REF, MEK, ERK, transcription factors. So there's all these, we have the phosphorylation of the guanosine exchange factor. Then that can activate the RAS phosphorylation, which then RAS can activate RAF, and RAF can activate MEK, and MEK can activate ERK, and ERK can activate a transcription factor, which then binds to activate the transcription. So these are long signaling transduction pathways that can go through there. One thing to think about is what can we do to mess around with this pathway? Think about what we could do. Why would we want to do that? Those are other things. So a couple of things that we could mess around with. We could block the autophosphorylation, so we can find something that binds here and doesn't allow the phosphorylation to occur, or you can mutate one of these things and put, if I mutate this phosphorylation site or this phosphorylation site, obviously we're not going to be able to activate or bind the GEF proteins. We can mutate or block any of, or put in inhibitors to any of these pathways where these compounds interact and associate. So we can do a lot of different things. And maybe, why would we want to? Well, maybe this pathway is not good, or we don't want this on in certain cells. So how, those are all things that we can do to target that. So this is another example of a different pathway. So you can see this is actually a different receptor. You have a different, this is a stem cell factor, and you have a different receiver receptor. It's called the KIT receptor. And this is going to dimerize, the receptor dimerizes, phosphorylates, autophosphorylates, and then it brings together this SOS protein, guanosine exchange factor, that's going to interact with RAS, that phosphorylates that, activates it, which activates RAF, MEK, ERK, and then another set of transcription factors. So this would happen in a different cell, would have a different result of for example, the melanoblast-inducing transcription factor. Different genes get in here. So these have different receptors, but they can have similar components. So you might, if you look more into signaling cascades, a lot of times we'll see the RAS, RAF, MEK, ERK. This is part of that. But they're able to, depending on what else is happening, what else is activated in here, whether transcription factors are associated, they can activate different sets of transcription factors and genes within the cell based on these pathways and where they are. Another signaling pathway that is important to think about and involved in a lot of different functions in our body, cell death pathway. So it's really important for brain development. Some of the spaces between your fingers and toes actually go away. We absorb a tail during development. So there's a lot of places where actually we want cell death to happen, where we don't want to have tissue, or that tissue has gone away over time. So this can also be a really important pathway for cancer. So if this pathway is deactivated in cancer cells, then those cells don't end up killing themselves. They won't die. And so this pathway can be important or valuable for if we're able to activate this in cancer cells, then usually that will allow us to get rid of the cancer. But a lot of times cancers are mutated in this pathway, so they won't turn it on. So an example of the importance of this actually in development, if you knock this out, in caspase 9, this is the caspase 9 down here, you get abnormal brain development. So sometimes that can change the developmental pattern. So you can see this forebrain is much larger in the knockout mouse than it is for the wild type mouse. That leads to developmental abnormalities and problems with that mouse brain function. So I want to briefly go over some of the development of making or finding drugs, creating drugs, how they get to market. A lot of times drugs, the way that you find those things are to isolate and purify proteins that you're going to target. Let's say we're just talking about all of those different MEK, ERK, RAF, right? All of the receptors. If you're trying to maybe use an inhibitor to the receptor, you might want to kind of clone or look at something that looks similar to the hormone that you're looking at. So you can do that by looking at using high throughput screens. So you can either use random targets, or you can basically use software to model what that compound looks like. So you can look at the structure. That's the smart design. So you could, maybe you want to block a pore, you want to find a receptor and kind of dock it to that. So then you can use what you know about the structure and shape and think about the intermolecular forces that might allow your drug to kind of bind to that and block whatever the normal function is. You can either do that kind of blindly, just looking at high throughput, just any drug that has ever been made, or any thing that you have that you want to throw on. You can kind of try to attach it and look at the binding affinities, or you can do the smart design techniques. This is a bit dated now, but the market shares, or how much we're talking about here in terms of the amount of money that's spent on billions of dollars. Most of them are monoclonal antibodies. Cytokines, for example, next to it, growth factors, hormones, enzymes, recombinant vaccines. Those are involved in here. Hormones here. Most of these are involved in hormones and growth factors, things that, well, those are essentially the same. That is most of the market share that we're talking about in terms of what things are generated and made. So the way that this gets to market, the way that we develop drugs is first we need to understand the system. So that's what you're doing right now, right? So looking at this, all of the different pathways, the basic research and understanding which receptor, which hormone is involved in whatever it is that you're looking at curing or treating. The next step is to then develop something, use that information to develop something that might be able to bind your receptor. So then you've developed something you think is going to work. The next step is translational research. So now, does this happen not just in the lab, but can we translate that into the clinic? And then we can do some more applied research. So once we get that into the clinic, what else do we have to do to improve? Are there other things that we have to use or do, or treatment options that we can look at? We also have to research that translational component. So what have we done? What are the results? How can we improve this? Most of this upfront section, the basic research, is happening in government and academic labs. There's some more of this translational research happening in government and private, non-private companies now, but most of the applied research is happening in biomedical research companies. So Pfizer, Merck, the companies that are making the drugs, their purpose is to create things to sell. They're not necessarily so interested in the basic research. They mostly get their information from government labs or academic labs that are funded generally by the federal government. So these are just kind of things, what is actually happening in this basic research just defines a little better. Looking at finding compounds, refining the drug, making it more stable, making a better binder. The translational is looking at the formula, developing pilots for manufacturing, looking at safety, efficacy, so that drug doesn't cause a lot of bad side effects. And then applied research is more, how do you manufacture it? Where do you subcontract it? How do you get market the drug? How do you sell it, distribute it? So these are all kind of part of this next step, that kind of subcategories of how this works. So I mentioned earlier you first have to find, you have to do enough basic research to understand what you're even looking for, or the pathway you're looking at, and then you want to target it. Find some compounds, do drug screens, or design a smart way to look at the structure of whatever it is, whatever pathway or protein you're trying to target. Find a lead compound, see if you can refine it and make it better, improve it, change a little bit, test some variables. What concentration do you need to use? And then eventually you need to think about getting it to human clinical trials, and then approve it for selling and marketing. In the early end of things, developing a model system is the most important part. A lot of times, for example, for Alzheimer's disease, you can't just start asking people to take random drugs to see if it works. Not okay, it's not ethical. But creating a model system is really important that is reproducible and models whatever it is, the disease that you're looking for. So is it well defined? Is it well characterized? The easiest model systems are those which are genetic and we know what it's causing that disease. But sometimes they're not that easy. Sometimes they're sporadic diseases, they're non-genetic, and we don't really know how. It's more difficult to study those and to create a model system for those. Are there good diagnostic biomarkers? Can we identify if there is a disease, or if there's some way to truly say if someone has Alzheimer's disease or not? How do we define that, or how do we know? And are there solutions that are available? So cancer, for example, can sometimes be screened from the blood or take a simple biopsy, but Alzheimer, it's not as easy as that. So, maybe part of what you need to do is just have, before you can even start making drugs or targeting drugs, you have to define the system. What is it that's really wrong, and how do you define if someone has that disease or not? A lot of times there are things that can be done especially for large drug screens. If you have a system, let's say we have a cell culture model that's modeling some type of inflammation, and we can find you can a lot of times use naturally occurring compounds or other drugs that are being used in other instances, and we can identify drugs that sort of work. And then you can use intelligent design versus high throughput screening, but you can modify those and make them better and change them slightly, change the organic chemistry of that makeup. Example of really helpful and useful remedy was penicillin. So basically, but then how do you find the active compound from that extract? You want to purify it. You want to maybe change a little bit to make it better. That kind of works. Repurpose things, for example, for angina, so a chest pain, a drug that was developed to was causing by the reduce of blood flow. They found actually that a drug used to increase blood flow to the heart actually increased blood flow to another part of the male anatomy that is now known as Viagra. So sometimes drugs can be repurposed. They're meant for one thing, but then ended up being valuable and for another thing. So when we go through the discovery, you want to target that compound, figure out where is it binding, how much time is it on it, how long does it metabolize for, or gets catabolized, when does it break down. So there's a lot of information that is involved in the discovery part. Then it's the clinical trials. So does it work? Is it safe? What's the dose range? How do you deliver the drug? How effective is it? And then there's clinical development, phase 1, 2, and 3 trials, looking again at safety and efficacy. How well does it work? How much gets into where it needs to go? Does it need to get into your brain? Does any of it get there? Really targeting the dose. And then once you get through phase 3 clinical trials, then it's generally approved and you get some chance to market this before other people are able to get in and make the generic drugs. One of the things that is important to figure out in terms of drug development is this IC50. So the kind of looking at this therapeutic index. So for animals, it's the lethal dose of 50. So that's 50% of the animals died versus the effective dose, ED50. So you look at the concentrations, usually in micromolar or millimolar concentrations of that compound. Obviously, we don't wait until we kill half of the people. So this is the initial screen. We do this in people or in animals to figure out that lethal dose. But in humans, you just want to go to the toxic dose. So when you start seeing side effects, that's when you stop, not above that. And then the effective dose. So the therapeutic index tells us how effective that drug is and how good it is. So you want, obviously, your effective dose to be a really, really tiny number, and you want your toxic dose to be a really, really big number. So this value should be really big. The larger your therapeutic index, the better the drug is. One other thing I should mention, the IC50 tells us where about half of the compound is bound to whatever the target is. So it's kind of looking at 100% binding, 0% binding, where is about half of it bound. And that gives us an indication of how effective that inhibitor is, or whatever it is that's going to bind the target. So again, you want that smaller number for the IC50 makes it a better drug. This is just looking at, again, the therapeutic index. For looking at mice, you have 100% of them living. You start giving this drug, and then we get down to 50% here, 0% once we get to 200 nanomolars of this drug of interest. The effective dose, so when do we start seeing happy mice? So 0% happy mice, they're sick, they're not happy. This is 100% happy. So once we get to 200, they're happy, but they're all dead. so that's not good. 50% here is, so if we throw in some numbers here, we have essentially this initial one is about 140 nanometers or nanomolar, and here we have about 60 nanomolar. So we have the lethal dose is at about 140. The effective dose is about 60. So again, that bigger, the higher the therapeutic index, then the higher that number, the better that drug is. So the cost of developing a drug is estimated at around $1.2 billion. So that is why the FDA generally gives 12 years of patent protection after the FDA approval happens, because they're trying to recover the cost of the $1 billion they spent up to that point. So it allows for, and then if you can find a different indicator or a different use for that drug, then you can extend it on to that 12 years of patent protection. Then works for that other indication. For example, like I mentioned, that heart pressure drug that then was used in Viagra. So they had another 12 years to use that in that other indication. So a lot of these, you might start with 10 or 100,000 compounds. And then you get to, you find the drugs, you maybe have 250 that you've kind of tweaked, and those are kind of preclinical. You're improving. This is the basic research. You maybe have 5 lead compounds that you go through the clinical trials, you go through the different amounts, and then eventually you'd find, one compound, 1 or 2 compounds. FDA reviews that and then approves that one drug for that treatment. So what do you think? Good drug. Some of the things that are barriers for this are, what's the population it's going to serve? If you're going to serve 2 people, well, the drug company is not going to be able to recover that $1 billion. So the size of the population is important. How often does it need to be dosed? And it depends on good. What do you mean by good? So is it good for the biotech company or for the patient? Obviously, 1 dose would be the best for the patient. The drug company, if they have to dose you every day, well, they can make more money that way. Cost of production, that's going to be important. Barriers to entry is another drug already have the market under wraps. Is that more hard to get into that market? Different treatment costs. How stable? What's the shelf life of it? Does it last for 10 years on the shelf, or do you have to make it every single day fresh? That's going to be an issue. And then oversight for treatment. Is it over the counter, or does it involve injecting into a certain part of the body or something like that? So that these all will change the effectiveness of a drug. So that's about it. I think the last few slides, even here, this last section was just kind of for self-interest, but that is all I have for the drug development and signaling pathways.